But instead, I want to welcome Marco Cecchini from the chemistry department in Strasbourg. And Marco is going to talk about molecular dynamics simulations of very large protein assemblies.
Thank you very much for the invitation. It's a great pleasure for me to be here at the Orap meeting and to show how we can use a fair amount of computed time to tackle a biological problem, and in particular study the function and regulation of important neurotransmitter receptors, which is depicted here in the slide. So as an outline of my talk, I will try to introduce you to the structure and function of ligand-gated ion channels, which are very important neurotransmitter receptors. And then I will discuss about our simulation approach to ion channel activation, which is a very important physiological process. And then I will discuss some of the mechanistic insights that we can grab from simulations, and which could not have been possible without the use of tier 0 resources. Okay, but if you want to provide with a physiological context to the problem that I want to investigate, then we should start with neurons. So neurons are excitable cells in the nervous system which process and transmit information. And for doing that, they have to communicate. And actually, an effective communication between neurons is vital to any organism and mediates fundamental cognitive processes such as learning, attention, and memory. And so the system that I'm investigating is mediate this communication at specific terminals, which are called synapses, which you see here. So in short, when an action potential invades the presynaptic terminal, neurotransmitter molecules are released in the synaptic cleft via exocytosis. So these molecules, which are depicted here as green dots, they diffuse into the synaptic cleft and bind to neurotransmitter receptors which sit on the postsynaptic membrane. And so in response to binding these receptors, they open an ion pore, which allows the flux of ions, so cations or anions, through the membrane. And this actually converts a chemical input, which is the increase of the local concentration of neurotransmitter, into an electrical output, which allows the transmission of the action potential. So these neurotransmitter receptors, if we look at the single molecule level, they are oligomeric transmembrane proteins, which expose one or more binding sites to the neurotransmitter on the extracellular side of the cell, and they form axial, cationic, or anionic channels, which are perpendicular to the membrane. So there are multiple states in which you can find these receptors, and so at rest, this channel is closed and no ion can go through. And in response to binding of neurotransmitters, so the signaling molecule, then there's a rapid conformational isomerization of the protein, which allows the flux of ions, so cations and ions, at rates that are approaching tens of millions of ions per second. So it's really a current. And so the focus of my talk is really this transition from a non-active to an active state, which is commonly referred to getting. And in the following of my presentation, I will keep my attention to the pentameric family, which includes, among others, the nicotinic acetylcholine receptor and the GABA receptor that are very important pharmacological targets in humans. Okay, so if we want to study getting with atomic resolution, we need to have structures. The first structure at that resolution of a pentameric ligand-gated ion channel was provided in 2005 by Nigel Unwin, based on cryo-electron microscopy analysis of nicotinic acetylcholine receptor-rich membranes from torpedo fish. And so despite the collection of this data was at modest resolution, so it was below 4 angstrom, this structure was actually quite instrumental to understand the relationship between structure and function in this very important family of receptors. And so in particular, this structure shows what is the overall architecture of the molecule, with a 5-fold axis of symmetry, which is perpendicular to the membrane. It shows the heteromeric structure of the protein, and also the presence of 3 domains. So one large extracellular domain, which carries the neurotransmitter binding site, which happens to be at the interface between subunits. Then a transmembrane domain here, which forms this axial channel, and a smaller but important intracellular domain, which is thought to be important for regulation of function of these channels. However, due to the modest resolution, it was not possible to assign to this specific structure a physiological state of the channel. The situation changed dramatically a few years later, so in 2008 and 2009, with the discovery of prokaryotic homologs of these channels in bacteria, which of course facilitated the expression, the purification, and eventually the crystallization. And so in 2008, the first crystal structure of a closed channel state was obtained by Ramon Dutzler in Zurich, and 1 year later, for another channel from Globacter vivacius called Glick, was obtained in the open structure. And even more, in 2011, so a couple of years later, there was the appearance of another structure from C. elegans, so this time from a eukaryotic organism, which was crystallized by Eric Guo and co-workers at Volume Institute. And so this structure is very remarkable for several reasons. And the first one is that, first of all, it's the first high-resolution representation of a eukaryotic channel in this family. And second, it was captured in the open channel state, so the active form, in the presence of ligands. And so now we can really localize with that resolution the position of ligands, and in particular, it was captured with the endogenous neurotransmitter bound here at the interface between subunits, which is L-glutamate, whose chemical structure is depicted here, and another ligand, which is a positive agonist, which is called ivermectin, which also binds at the interface between subunit, but this time in the transmembrane domain. So this structure, together with the next structure that was crystallized 3 years later, which was solved in the absence of ligand of the same channel, BlueCl, offers the first high-resolution description of the Gettin isomerization. So we can have the two high-resolution structure of the endpoints of this important transition, which is the focus of my analysis. And so in particular, if we compare the structures, so the resting state and the active state structure of the receptor, we can notice already differences. And for instance, in the absence of ligands, the ion channel shuts in the middle, so at the position which is called 9'. So also the comparison shows that important arrangement must occur at the interface between the extracellular domain and the transmembrane domain, and also that there's a global change of the extracellular domain going from resting to active. And actually what this structure shows is that in the active state, the extracellular domain is significantly more contracted and becomes more relaxed or expanded when it goes to the resting state. So with this structural information at hand, what we want to do is understanding the details of Gettin by running all-atom molecular dynamics, and eventually trying to understand what is the allosteric coupling between the binding of neurotransmitter, which occurs up here, and the opening closing of the ion pore, which is down here, at a distance that is 60 angstrom away. So it's a big distance for molecules. And we want to do this with molecular dynamics. Okay, so what is our strategy? So our strategy is very simple. What we want to do is to trigger a spontaneous isomerization of the channel in one direction or the other and watch it live through molecular dynamics. So this sounds like impossible, and so how we can do that? And so if we think of the free energy surface that is experienced by the channel during the isomerization, we can think that actually this free energy surface, there are two free energy surfaces. So one that corresponds to the bound state in the presence of ligands, of agonists, which stabilizes the open channel state, and another free energy surface, which is the one of the unbound state, which actually favors the closed channel state. So in this interpretation, if we start with the active state of the receptor in the presence of ligand and we equilibrate it in molecular dynamics, we can actually think of promoting a vertical excitation to the unbound free energy surface simply by removing the ligands. And that is actually what we want to do. And so if we take then the structure in the absence of ligand and we simulate it long enough, and so we allow the time to relax to the structure, hopefully we can see the relaxation, spontaneous relaxation to the closed state, and learn something about Gettin. Okay, so computationally, this corresponds to taking the structure of BlueCl so the glutamate-gated chloride channel, in its active and resting state, so preparing full atomistic models for these two structures, embedding the structures in a pre-equilibrated lipid bilayers, adding the solvent and the ions on top and the bottom of the simulation box, deciding on the protonation state of the isonazole residues, because in molecular dynamics you cannot change dynamically the protonation state, then finding parameters for the ligands, and in particular ivermectin, and then running molecular dynamics at constant temperature, so 300 Kelvin, and pressure, so 1 bar of pressure. And we do that using AMD. Okay, so to just understand or let you appreciate the complexity of the problem that we want to tackle, I had an animation, which I think is not working at the moment, but it doesn't matter. So the only thing I want to say is that for those of you who are not familiar with molecular dynamics, so running simulation for a system which accounts for about 200,000 atoms is computationally very intensive, because we have to solve the Newton's equation of motion for each individual atom of our simulation box. And so, first of all, as you can see from this picture here, where the protein is depicted in yellow, the membrane is depicted in cyan, then the solvent is in red, and the ions are in yellow and green. So there are lots of centers, and so the system is huge. And second problem is that once we have the molecular dynamics trajectories, we have a series of snapshots of a huge number of degrees of freedom. And so we need to rationalize this information, this overwhelming information, or this overwhelming complexity. So we need to identify specific structural observable, or what we call in the field reaction coordinate, to understand what is going on. Moreover, if we talk about computational performances, which I think is very relevant for this forum here, so if we take a system like this, a biological system with 200,000 atoms on a cluster of 512 CPU cores, which is approximately the size of the cluster that I have in my own lab, I can run simulation with a performance of 6 nanoseconds a day. And 6 nanoseconds a day is nothing relative to the biological timescales, which are on the millisecond timescales. So clearly, if I want to run the simulation properly, I need to have lots of computer time, so lots of computer resources. And of course, the situation is different if we can run on tier 0 resources. In fact, as we have demonstrated here, so using optimized or in-house optimized codes and first-class computer resources like those that we can access to the Curie thin nodes, then if we run on about 3,500 cores, we can get performances which are about 60 nanoseconds a day. So before it was 6, now it's 60 nanoseconds a day. We can easily gain a factor of 10, which is very, very important, because in the same amount of computer time with these resources, I can reach microsecond sampling, which is maybe not enough to cover the full biological scale, but is very important already. And I can do multiple repeats, which is extremely important to collect statistically meaningful results. So the only thing that I also want to point out is that there's a cost to be paid, and actually 1 microsecond sampling, which is my target several times, approximately costs 1.4 million CPU hours, which is quite significant, and requires uncommon computer resources like those that are typically offered in the PRA's competitions. And in fact, to study the getting in GLUCL, so we profited from a 20 million hours allocation that we got at the PRA's level, and so we decided to set out, starting from the crystal structures, the resolution structure of the active and resting state, and performing the following experiments. So there are two kinds of experiments that we perform. So the first one is simulating the stable states, so the active state of the channel in the presence of ligand, and the resting state of the channel in the absence of ligand, for something like sub-microsecond trajectories, so 500 nanoseconds and 200 nanoseconds. Why we do so? Well, first of all, because we want to verify that the simulation setup is appropriate, so that we have a certain stability of the structures in simulations, and also because these simulations are quite instrumental to collect what we call the collective variables, so the reaction coordinates. So observables that can distinguish between active and resting. And then using these variables to actually question the true experiments, which are the two relaxation of the GLUCL channel in which we selectively remove the allosteric agonist ivermectin. And these simulations were run for about 2.5 microseconds, which are very extended simulations and are independent runs. So that's the plan. Let's take a look at the results that we can collect using simulations. So the first striking observation is that if you look at the relaxation, the microsecond relaxation of the channel, well, in both cases, we can see a spontaneous closing of the ion channel. So in fact, if we monitor the cross-section of the ion pore as a function of time, we can see that the two simulations here, which are in blue and light blue, they start with values which are compatible with an active state, which is in red here, so an open channel state. And after removal of ivermectin, with time, they evolve to values which are consistent with the resting state, with a closed channel state. So these simulations suggest or indicate that actually the spontaneous closing, so that the closing of the ion pore is spontaneous in the absence of ligand. They also say that the closing is a slow process, which takes about 400 nanoseconds in the fastest case, and also that this closing is robust to the initial simulation conditions, because we can sample in two independent runs. Even more striking, if we monitor the C-alpha rhythmic square deviation of the transmembrane domain, so where the ion channel is, relative to the crystal structure of the resting state, so that was solved in the absence of ligand, we can see that actually the transmembrane domain, upon removal of ivermectin, converges to the crystal structure, with a final rhythmic square deviation that is about 1.4 angstrom from the experimental result, which is quite consistent with the temperature fluctuations in the simulations, and actually with an ion pore that shuts, I have here, at position 9', which is in perfect agreement with the experimental result. So from my perspective, these results are quite significant because they indicate that molecular dynamics started with the active state structure in the absence of ligand, were able to produce the experimental structure solved in the absence of ligand, which to me suggests that molecular dynamics can be predicted. So the second striking observation that we have made is that the relaxation of the channel, and so the closing of the channel, is not a local structural event, but involves a global reorganization of the protein. And in fact, if we monitor the relative position of the actual cell domain relative to the transmembrane domain, we can clearly see that during the trajectory, there's a quite large twisting transition, so from value starting from 12 that goes to about 22, and so the global receptor twists in response to ivermectin unbinding. And actually, what also the simulation suggests is that the twisting transition correlates with the closing of the ion pore, which implies that the twisting transition controls the opening and closing of the ion pore. Then a second observation that we have made is that during the relaxation upon removal of ivermectin, there is also a radial expansion of the active cell domain as described by the crystal structure. And in fact, if we monitor the polar tilting angle of the ectocellular subunit, what we call the Blumen, we see that the simulation starts with a value that is about 7, and then they move to a value that is quite consistent with the resting state, which corresponds to something like 12. So each individual subunit tilts upward in the radial direction, such that the overall ectocellular domain expands in the radial direction. And also what is important from, as is depicted here in the model, and also what is important from the simulation results is that it looks like that these two transitions, these two global transitions, are actually distinct. And then first you have to complete the twisting isomerization before activating the blooming isomerization and finally reach the physiologically resting state. So in order to show you this sequential character of the global transition of the protein, I have an animation which I hope is shown. And so let's see whether I can start it again. So at the beginning of the simulation, so I am depicting here the protein from the top view. So what we see in colors is the relaxation of the glucial in the absence of ivermectin, and what you will see in transparent is the initial coordinate of the active state, which is used as reference. And so if you look in the initial part of the simulation, you have a clockwise twisting of the subunits until they reach a point in which the subunits start to get decoupled, and so they expand in the radial direction. Yes, unfortunately, the movie is short, and let's see whether... Okay, so what happened? Okay, so we can watch it once more. So first, the subunits, they run in the clockwise direction, and then they expand like the blooming of a flower. Okay, so the question is, the spontaneous closing is not a local event, it's a global event, but why is a global event? So what is the functional role of this large amplitude movement of the protein? And so now it comes a third striking observation that we made, and is that essentially this global movement of the protein, they occur with significant restructuring of the interfaces between subunits. And these interfaces are very important because those are the binding sites for the regulatory ligands. And in fact, if we compare, for example, the binding site of ivermectin, which binds in the transmembrane domain, and take a snapshot before and after the twisting isomerization, it is quite clear that twisting produces a significant movement of residues at this level, so that it shrinks the ivermectin binding pocket, which at least hinders the binding affinity, if not kills completely the binding affinity of ivermectin. And the similar thing is true if you look at the orthosteric site with endogenous neurotransmitter binds, because if we compare snapshots taken before and after the blooming isomerization, so the second part of the transition, we can see that we have a significant expansion of the orthosteric site due to the expansion of the extracellular domain, which of course reduces the binding affinity for the neurotransmitter. So overall, what the simulation suggests is that the global isomerization which controls gating occurs with significant restructuring of the subunit interfaces where the ligands bind. And so this movement can be controlled by ligand binding, and so ligand binding can then be coupled to the opening closing of the ion pore. So this global movement of the protein, which are encoded in the structure of the protein, they provide this allosteric coupling. So as the take-home messages, the fair amount of computer time that we used, that we invested in the project, and also thanks to the P-race allocation, provided the first high-resolution description of the reverse of gating, so the conformational transition from the resting to the active state of a pentameric ligand-gated ion channel, which provides interesting mechanistic insight. And so the simulation suggests that channel deactivation is composed of at least two distinct isomerizations. So a twisting transition, which controls pore opening and closing, and the blooming isomerization, which regulates the transition to a sort of preactive intermediate state that I didn't have time to discuss about. And also, the simulation provides a plausible model for the allosteric coupling between neurotransmitter binding and ion pore opening 6 angstroms away, which helps rationalizing the effect of positive allosteric modulators, like ivermectin, in ligand-gated ion channels, and eventually can be exploited to design new modulatory ligands. And so all these results, they've just been accepted in PLOS computational biology. So before concluding, let me thank the people who contributed to this work. So Nicolas Calimet and Nicolas Martin, so a PhD and a postdoc student in my lab, and 2 more senior advisors, so Martin Karplus from Harvard University and University of Strasbourg, and Jean-Pierre Changer at SCV Pasteur, who was basically one of the founding fathers of protein allostery. And of course, last but not least, let me thank all the agencies which provided us with the computer time that was required to run the simulations and to get this mechanistic information. And thank you very much for your attention.
Thank you, Marco, for this very interesting view of an incredible molecular machine. I guess probably people know that Martin Karplus got the 2013 Chemistry Nobel Prize, and also that the electron microscopy technology got this year's Chemistry Nobel Prize last week. And hopefully Jean-Pierre Changer is a future Nobel Prize. And
That's what we hope.
who knows? So thank you, Marco. Questions on this somewhat more biological topic? Yes.
Are there pharmacological applications yet foreseen
Of course, there are many pharmacological applications, and not on the system that we have studied, because that is from C. elegans, so I don't think that the pharmacology for C. elegans is very important. But what is key, and I think is one of the reasons why cryo-electron microscopy got the Nobel Prize this year, is that actually there are more and more structures coming out from human, for example, so glycine receptor, GABA receptor. There's one structure of the nicotinic acetylcholine receptors, so now we can use X-ray crystallography and cryo-EM microscopy to get more and more structure of pharmacologically relevant receptors, and now we can really do simulations on important targets. And so I think, what we wanted to do here was to use at maximum the structure information that was available at the time and sort of producing a model of the transition. I don't see why the new receptors, which are important in humans, cannot have a similar transition or cannot be studied with similar techniques. Of course, we need lots of computer time, right? And what one can think now, and is actually what we're doing in the lab, is now we have identified the movements which are important. Now you can pull these movements and then maybe compute in free energies along these movements. And so instead of waiting for spontaneous transitions, you can force the transitions in the presence of different ligands and see what is the effect in the free energy surface.
Just a comment, I think it's interesting that the structure appeared in 2014, and so then you really have a throughput issue because there's competition going on, and from the moment when that structure becomes available, you need to be fast enough to actually publish.
Yeah. In fact,
First.
in our analysis, we didn't really use that structure. The structure was used only as a reference. We didn't put this information that was the one of the resting state. So we started before. But it is true that, structures are, the competition is very tough.
Okay, evidently you were not scooped, though. Probably Praise had something to do with that. So, Stefan?
There was a few years ago a discussion and debate about a dedicated machine like Anton machine in your domain. What is now the current state of the art? Is it Antoning still present? What are your view regarding exascale challenges? Do you plan to use general purpose machine, even if they are mini core or GPU based? Or do you think that Anton is still a track to continue?
Well, Anton has got a big advantage over the others at the moment, and I think, what you said before, that is having dedicated machines for a specific problem is really the key if you really want to succeed in a specific field. And exa scale computer resources will help us for sure, but I think there must be a development in both directions, so both in the amount of computer time that will be available to us and also the actual speed of these calculations. We also run simulations on GPUs, but the problem of GPUs at the moment is that they're good relative to CPUs, but for the short amount of 6 nanoseconds a day, so for short simulations. You cannot use the GPUs to get the 60 nanoseconds a day when you scale on 3,500 CPUs. So at that time, GPUs were not an option for us.
Okay, one last question.
Do you understand the significance of the twisting in the whole process?
Yes. I didn't have time to go through, and also because it is mechanistically complex. And so, just in a few words, what happens is that the twisting isomerization controls the position of two specific residues at the interface. So, if I can use my hands, so I'm Italian, so I like to use my hands, right? So, you have two residues at the interface, and there's a proline, a totally conserved proline, which is on the outside of the channel when the channel is open, that wants to go through to close the channel. And there's one specific residue here which is making a barrier. So the twisting is shifting these two residues in such a way that when you twist significantly, then the proline can go through and close the channel. So our model is that basically the active state of the receptor in the absence of ligand is a high energy state. So there's some strain into these configurations. And so the only thing it can do is first twist in, and once it twists, so the program can go through and close the pore. So that's why I said that twisting controls pore closing.
Thank you, Marco. I'd like to thank all the speakers of this session for some very interesting talks. Thank you all.